Hazel Barton is currently a professor and director of the Integrated Bioscience Program at the University of Akron. She is an incredibly accomplished scientist and leader both inside and outside the caving community. In addition to researching the microbiology of Lechuguilla Cave, she has also contributed to the exploration of the cave. If you all were able to tune in to the exploration session yesterday, you hopefully caught her presentation, The Mazes of Lechuguilla Cave, Chandelier, Graveyard, and the Voids. With that, I will go ahead and hand the presentation over to you, Hazel. I can see your slide. It looks great. And your pointer.
Thanks, everyone. I know we're getting to the end and it's been a long weekend and there's been a lot to take in. So I appreciate it for those of you that have stuck with it this far. Hopefully I did actually not pick the end to kind of like have the ending talk, but so much that we could shuffle the schedule if I needed to jump around. I was going to be a bit more flexible, but I still ended up at the end because everybody was able to fit their talks in. So thanks to everybody for that. I'm going to talk about my work in cave microbiology, and most of you probably know that I was a postdoc with Norm Pace. So Norm hired me because he told me that he likes the way that cavers think, and I ended up working with him for a couple of years. My background and training is in medical microbiology, but Norm trained me in cave microbiology. And Norm actually hired me to work on tuberculosis. And tuberculosis is a pretty devastating disease. About 300 million people worldwide are infected with it. It's one of the biggest killers in the world right now. Without treatment, you will die from it. And he was interested in looking at the microbiology of tuberculosis infections in the lung. And you don't want to do that with humans, because if you're around humans who are sick, then there's a likelihood that you might get sick from them. So I worked on an animal model, and that was a guinea pig. And the reason we work on guinea pigs is that they can't cough. So you've probably all seen the images of people gowned up for working with COVID. Well, I would have to dress up like that to go in and infect these poor guinea pigs and then study the microbiology of their lungs. One of the things that turned out to be a huge problem with this microbiology project is that guinea pigs have tiny lungs. They're about the size of a thumbnail. And the lesions that you get from tuberculosis they're made from a calcium oxide. And the immune system, it seals off the infection to protect the body, but it does that with calcium. And calcium sticks to DNA really tightly. And so it's very difficult to separate the DNA from the calcium. And as a result, you really have a hard time doing microbiology. We don't really culture anymore. 99 depending on which system you use, let's say 99% of everything that we know is out there is unculturable. And so that's where Diana was showing you the data based on DNA. So what we do is extract DNA from the environment, and we use a kind of barcoding to identify the unique microorganisms. And that's what Norm and I are pointing at in my notebook there. We do it high throughput now, but that was back in the day when it had to all be done by hand. So what I did get very good at is not figuring out the tuberculosis model, but extracting DNA from these calcium-rich environments. And as a caver, that kind of appealed to me. So I asked Norm if I could take this technique into a cave, as the tuberculosis project wasn't working out. And of course, him being a caver, I had his full support. So I ended up doing some early cave studies. This is a study, I'm getting help from Michael Queen here. This was actually in Carlsbad Caverns. But my very first project was actually in Glenwood Caverns in Colorado. And I was exploring it at the time. I was the cartographer for the cave, or am the cartography for the cave. I'm not sure if I am anyway. And we just discovered this brand new area that held all this biofilm material on the one side, and it'd been fossilized, and we called it the Astrobiolab because it looked pretty funky. And I was on the discovery and I knew it was pristine. So I thought I could go in and have a look at the microbiology living on the walls. And what was different about this kind of microbiology is we weren't looking for snots and slimes and things like that. I was going up and just taking just a regular wall. So just as you would look at maybe the microbiology on the wall in your house, you really don't see anything. you've probably got 10 to 1,000 times more microbes living on the paint on your wall than you find in a cave. So I used this technique that I developed for the tuberculosis project. I applied it to the cave, and it turned out that we discovered an incredibly diverse array of microorganisms. And what made this significant is that we thought going into this really starved cave where there was no energy, there was no sunlight, there really wasn't any moisture percolating in from the surface, that we'd find just a few scant bacteria that was specialized for those really starved conditions. And it ended up being incredibly diverse. I think in that study, which was pretty crude back then, we identified 13 different phyla. So for 13 different kingdom levels, different groups within the bacteria. So that basically has been what I've spent the rest of my microbial career trying to understand, is how microorganisms can be so diverse in caves under such nutrient limited conditions. And there is precedent for this in the literature. Hutchinson in 1960s described something he called the paradox of the plankton. And that is where a limited range of resources actually supports a much wider range of diversity than would be expected. And his data is looking at different ponds, and the more nutrient limited, the less energy, the less food stuff going into the pond, the more you would actually see diversity within the plankton. And so I've started using that literature to try and understand how microorganisms are diverse in the environment. So I'm going to summarize about 20 years of work now with one slide. So this is kind of my politics slide, right? So the thing about politics is you have two very different ideas of how you solve a problem. So the problem is, how do you make a living in extreme starvation? And so there are two approaches you can take. One is cooperation. You all work together to get the resources you need to survive. And the other is competition, where you go in and you basically, kill your competition and either suck the resources out of them or take advantage of the environment that they were living in. For the cooperation part of this, I've actually been working in Wind Cave in South Dakota. So I've been studying the microbiology of the Wind Cave lakes. This is, the lakes in Wind Cave are very different. The only lake in Lechuguilla that would be comparable is Lake of the White Roses. It's the similar kind of thing where the cave intersects the aquifer. So this is not a basin. So if you think about virtually, well, all but Lake of the White Roses and Lechuguilla, these are basins that water drips into. Wind Cave actually intersects the Madison Aquifer itself. So you have a lot of microbiology and hydrology, and it's a mixing zone going on there. It's also got the lowest cell density of any water on Earth. So we were looking at, it's about 2.3 thousand cells per mil, which is seawater, if you can see through it, it's about 1 million cells per mil. So we're about 2,000. The only place that's gotten close to this are very deep South African gold mines, about 2 kilometers underground. It's been really hard to purify enough bacteria to do the work that we've needed to do, but we've actually worked on using these kind of dialysis systems. This is a tangential flow filter that is actually used for doing kidney dialysis, and it has a pore size of 40 nanometers. So I'm trying to, what's a good way of describing 40 nanometers? So if you take one of the hairs from your head, it's about 100 micrometers. So it's about 40,000 times narrower than a hair on your head. So very, very tiny holes. And this work has been really productive. We've been able to show this very high diversity with this low biomass. We've collaborated with a couple of groups and identified that these organisms are using unique metabolisms that are pretty ancient. And they also are in this new group. Diana was talking about the big tree of life. So there's this new group of bacteria that's been isolated. It's called the candidate Phylum radiation, which is kind of a technical term for a bunch of kingdom level divisions within the bacteria that's just kind of off to the side of the classic ancestral grouping of the bacteria. This may end up being a fourth domain of life. We're not sure yet. The reason that they've only just recently been discovered is those barcodes that we use to identify bacteria don't really work in this group. You can only identify them using molecular sequencing of the genome. And they're ultra small. So these things are very tiny. Almost all of them are below the theoretical limit of life. So most people miss them in their environmental surveys because they filter with 0.1 micron filter. So that's what 1,000th the width of a human hair. And these guys will slip right through that. It turns out that the wing cave lakes have about 40% of the population within this group here. And we've been studying how they metabolize and live in that environment using single cell whole genome sequencing. So what we do is actually isolate individual cells and sequence the genome of those individual cells to figure out how they're growing, because not one of this group has ever been cultured. And that's in collaboration with the Joint Genome Institute. And we actually, I was hoping I could put it up here. We've got a Nature Microbiology paper coming out tomorrow, but I'm not allowed to say anything until they have the press release. Anyway, we're supposed to be talking about Lechuguilla, so I'm not going to spend too much more time on wind cave. But we did take the technologies that we've used in wind cave and we have applied them in Lechuguilla. And at least in Lake Castrovalva, we don't see the diversity that we see in wind cave. So as I said, these are incredibly oligotrophic environment in wind cave where we see, really phenomenal diversity, 26 different phylum. At least in Lake Castrovalva, where we did the similar techniques. So we isolated about 100 liters of water and purified the bacteria. We sequenced about 4 million tags. We didn't see the same level of diversity. It was somewhere between about 12 and 15% of what we see at Wind Cave. That's not to say that the lakes at Lechuguilla aren't diverse, but I think these pool basins that are calcified don't have that kind of microbiology that perhaps we would see at Lake of the White Roses, and we certainly need to go there and check that out at some point if time permits. Anyway, so Lechuguilla actually comes under the banner of competition. So I'm using wind for cooperation and Lechuguilla is for competition. And the reason Lechuguilla works so well for competition is it's a very deep and isolated cave. As you've heard many times, it's got a single entrance. It also has a sandstone caprock, and that sandstone caprock stops water coming in. It takes a long time to come in. And as you saw from Jake's presentation earlier, they aren't detecting water from the bomb blasts below about 200 meters. So this suggests that it's, the percolation time from the surface down into the deep level of the caves might take 100 years. And because of that, it means that we can sample below that zone and actually look at microbial communities that haven't really been influenced by human activities. I was given a presentation on Lechuguilla and talking about the microbiology of Lechuguilla at a conference. And this guy ran up, Jerry Wright, and asked me if I'd looked at antibiotic resistance in Lechuguilla. And I said, well, why would there be any antibiotic resistance in there? No one uses antibiotics in the cave. It may be that, somebody's taking antibiotics and might pee at one of the spots, but for the broad area of the cave, the cave's isolated from those antibiotics because the water takes so long to come in. But we went and did it anyway, and it turned out that there was a resistome, actually much higher than other environments that Jerry had looked at. So what we're looking at, we're using Lechuguilla Cave is basically an analog for a pre-antibiotic era environment. The work we did in there, along here, we have all the antibiotics that are currently used by doctors to treat you if you have a gram-positive infection. So that would be something like methicillin-resistant Staphylococcus aureus or MRSA. And then below would be gram-negative, so maybe treat you if you had a salmonella infection. And you can see in some cases we're above like 90% of the bacteria that we isolated in one sample being resistant to this particular antibiotic. It was also pretty interesting because they were resistant to some of the drugs of last resort, like daptomycin, which is thought to have no antibiotic resistance. It's thought that you can't develop it to it. And 20% of our isolates were actually resistant to daptomycin. So this was actually a pretty big paper because it's every other environment that people have looked at, it's possible to come up with scenarios where antibiotics could have been introduced, but we were collecting these samples in pristine environments where we knew people hadn't been. Why it was, I think, yeah, it was challenging the conventional wisdom, because when Fleming, if you're not familiar where antibiotics come from, they were isolated by Alexander Fleming. He was a British microbiologist, and he noticed that when a fungus grew on a plate, it was a species of penicillium, which is the blue mold on bread, it prevented the growth of bacteria. So you see this area where the bacteria can't grow near the fungus. And that's because the fungus was producing a compound called penicillin. And this became a huge part of the war effort to isolate this compound and use it to treat disease. And in his 1945 Nobel Prize acceptance speech, Fleming actually warned that we needed to be careful using antibiotics, because he'd been able to show in culture very easily that microorganisms were able to become resistant to sublethal levels of penicillin. So if you used it at a high dose that was enough to kill, then the bacteria would die. But if you used it at a low level for a long time, they would actually evolve to become resistant. And that's basically what's happened. Everybody's aware of the antibiotic resistance crisis. Right now, we're predicting that the annual death toll from infections that we would have been able to treat 20 years ago will be 10 million people. I'm sorry that this value is in pounds, but currently our government doesn't give statistics like this. But it's estimated that it'll cost between now and 2050, $66 trillion in medical expenses that we wouldn't otherwise need because of the development of antibiotic resistance. And to clarify what I mean by that, I mean that infection that could have been treated if you'd have gone in and that organism had not been resistant to the drugs that they're using. And microbiologists have been waving their arms in the air about this for, I don't know, I think I remember hearing about an antibiotic resistant crisis in the 90s. And, you know, people don't really care about microbes because, you know, they don't know anyone that's sick. Why would they worry about it? And of course, the COVID epidemic, pandemic has changed all that. And people are really starting to understand the significance of microbial infections. Obviously, COVID is a virus, but, you know, the death toll from COVID is not approached yet what we think we might get from antibiotic resistance. We did look, and so what was the outcome of the study in Lechuguilla? Well, we were able to say that antibiotic resistance is natural, ancient, and hardwired into the microbial pangenome. And it's ancient because the Lechuguilla cave has been isolated from the surface for so long, and certainly in the areas that we were testing. And this really changes the paradigm for how we think about resistance, because it means it's essentially out there everywhere. And we need to take into account resistance when we're developing drugs. So one of the other papers that we published is looking at one of those bugs. So one of the bugs that we isolated had this really weird phenotype where it didn't seem to be killed by anything. It's a Bacilli, so it won't make you sick. It's not a superbug. They're everywhere in the cave. They're pretty innocuous. Imagine coming home and seeing a puppy lying on a road kind of on its back with its legs in the air. That would be the metaphor for a Bacilli in the environment. It turned out that this is just a picture here showing you some of the different classes, the structures of antibiotics that are out there. It turned out that this one strain from Lechuguilla was resistant to 26 different antibiotics. That was resistant to 14 different classes. So that means 14 different structures or types of structure of antibiotic. And it was able to inactivate 7 classes of these antibiotics. Now, most resistance occurs by the bacterium actually changing its target so that when you throw an antibiotic at it, it's a miss. In this case, the bacterium actually made enzymes that would damage the antibiotic itself, so it was no longer functional. And it turns out that 3 of these actually were completely novel. They'd never been seen before. And I remember when this came out, I worked with the press release people at the park, and I'm like, okay, we need to handle this very, very carefully, because we don't want people thinking the superbug, the Andromeda strains just come out of Lechuguilla Cave. So this is one of the mechanisms of the resistance. This is a amido hydrolase. It basically cleaves an amino acid off of this antibiotic, which is called bacitracin. If you've ever used Neosporin, there's bacitracin in the Neosporin. And this is just showing the enzymatic, the functional domain of our protein binding to the antibiotic to do the cleavage. And when you do that, you basically knock out the ability of the antibiotic to have any effect. And this is just a picture. Well, what we've got is this is E. coli grown on a lawn of bacteria with a drop of bacitracin. And you can see the bacteria won't grow anywhere near the antibiotic. And then what we've done is we've taken the gene that encodes for this resistance and we've put it in E. coli, and you can see now it's happily resistant to the antibiotic. Now, when I talked to the park about the press release, I said what we need to do is we need to talk about what this means in a positive way. And what it means in a positive way is these mechanisms of resistance aren't out in the clinic right now. It means if you get an infection and there is resistance to it, it's not part of these methods that this bacteria produces in Lechuguilla Cave. So it might be 20 or 30 years before it emerges out of a different environmental strain, because it's not going to just be this strain in just this cave. It'll be out there in the environment. What we can do is we can actually develop something called an adjuvant. And what an adjuvant is a compound that will actually inactivate the inactivator. So if this is let's say this is the enzyme that inactivates the antibiotic, imagine we find a compound that can sit in this little pocket here so that the antibiotic resistance can't cleave the antibiotic. This is that very phenomenon. This is a plate of bacteria that are resistant to penicillin. So this disc, we have a tiny bit of penicillin here, and we add more and more and more till we have very, very high doses of penicillin. And you can see that the bacteria can grow very close to it. In this test here, they've done exactly the same thing, but they've added a drop of this compound called clavulanic acid. And what that does is actually inhibit the enzyme or the binding of the enzyme that causes resistance. And you can see that resistance is restored. And so we call these adjuvants because they basically destroy resistance. And if you've ever taken augmentin is a mixture of penicillin and clavulanic acid to do just this very thing. So, by the 1960s, 1970s, penicillin wasn't effective against a broad range of bacteria, but the addition of clavulanic acid has basically restored the activity of penicillin. So when we're in there thinking about resistance, the other thing we thought about, well, if there's a lot of antibiotic resistance, that means that something's selecting for it. So there must be a lot of antibiotics. So we went on an antibiotic hunt. This is one of my undergraduate students with some of the isolates from Lechuguilla. And on one of the plates, we identified this. And this is a crystal that you see the bacterial colony here. And you see the crystal. I got that, thanks, Pat. You see the crystal precipitating out. And it turned out that this bacterium, this is also a Paenibacilli, was making so much antibiotic that it was actually spontaneously crystallizing out of the media. And this is iodine. So this is an antimicrobial compound. It turns out you can't use this in humans. It's toxic to humans, but it suggested that there were lots of potential for antibiotics. So we started collaborating with this guy, Brian Brackman at Vanderbilt. And we did the same thing we did with Jerry. We took a small group of microbes from Lechuguilla and we looked for antibiotics, and we did find a novel antibiotic, which we called lechomycin. This doesn't have specific activity, so we can't use this as a drug. Everybody gets excited when they show this slide. And every time I show this slide, I remember I've got to finish writing the paper, and we do have a deadline at the end of the month. So as soon as this thing is over, I can start working on that paper. In 2007, Dale mentioned the benefit sharing agreement with the national park and how Carlsbad had set these up. And we actually, I was kind of the go-to person for this, but we actually set one up between Carlsbad Cavendish National Park and Cubis Pharmaceutical in 2007. And this thing, we got it in 2007. I think we started in 2005, because it was basically lawyers agreeing on if, you know, Cubis found $1 billion drug, how much would the park get? And we actually managed to get this thing through. And I thought that, you know, benefit sharing, these, you know, these are pretty common. Everybody knows what, you know, knows what these things are and everything. And the woman who ran the program actually told me it was the second one they'd ever successfully finished in the Park Service history. So that was pretty exciting. So the idea was we were going to do a massive screen for potential antibiotic compounds in Lechuguilla Cave. And then almost immediately afterwards, Cubis got bought out by Merck, and Merck shut down the entire operation. So natural product discovery ended in that path. But we've been doing a lot of work. This is pretty labor intensive, the traditional way we were doing it, which is where you pick an isolate, you get it on a plate, and then you isolate in pure culture. And we've started working with Brian Murphy at University of Chicago. And we've actually ended up with a high throughput system where we grow bacteria on a plate. And we can actually use a multi-tough. So what you would do is just pick individual bacterial colonies onto a plate like this and zap them with a zapper, which is, I'm trying to remember, mass, was it moldy? I know it's laser ablation. I know the chemistry behind it, but I'm tired. Anyway, it's moldy tough. You zap them with a laser, they get gassed off. You suck that gas through a time of flight mass spectrometer, and you can identify the bacterial species based on what gets liberated when they zapped. And you can also identify novel compounds doing this way. So we can actually identify novel metabolic products. And we use this technique to actually do high throughput. So instead of, you know, picking and laboriously checking, we can throw 200 on a single plate and then identify the 3 that we need to pursue. And we're also doing genomic sequencing. We've already published some of the genomes out of Lechuguilla. There's a new technique now called a mini ion, and this is basically a DNA sequencer that you can plug into the USB port of your computer, and you can run the DNA sequence. And we can do a genome for about $10 using this technique. And the goal is to sequence 1,000 genomes from Lechuguilla. We take that genomic information and we actually search for antibiotics. So it's a 2-step process, identify the ones we're interested in this way, and then sequence the genome. And I know it's called a mini ion, but I like to say minion, and I drive all my collaborators nuts because I call it the Minion Project and keep sending them pictures of minions. And then I know I'm running out of time, but I want to reiterate what everybody said about Kim Cunningham. I think he launched modern microbiology in caves, and he also recognized the significance of exploration. Exploration means that you can go into a pristine environment and know that you are the first person in there. And so that when you're studying the microorganisms, you know they haven't been impacted in ways which would confound the studies that we're trying to do. So we're able to follow the explorers when they make discoveries and do some culture libraries where we're actually, in this case, we actually went up the Alpine Twister and took a series of samples up the Alpine Twister into Oz to see how the distance from the surface, which is right here, affects the microbial community. And then we generated this very large library that we can screen using those techniques I described. Using this, we can screen about 100,000 colonies quite quickly. And so we've submitted a proposal to the National Institutes of Health to do this. And our goal is, if we're funded, to screen 1 million bacteria from Lechuguilla for novel antibiotics. sequenced 1,000 genomes and identified 10 novel antibiotics using this approach. And so to answer the question, which is driving competition, which is driving diversity in caves, is it cooperation or competition? And unfortunately, just like politics, it ends up being a combination of both. And I want to thank all my students. Olivia Hershey has been doing the work in Wind Cave. Katie Bender has been doing the work in Lechuguilla. My collaborators I've mentioned. And I know I'm out of time and they're texting me, telling me to stop. But there's so many people that I have to thank who've been Sherpas that if I were to do it, I would burn way too much time and not spend enough time talking about the science. So you guys know who you are. You know, I couldn't have done it without you. And a lot of what we've been able to accomplish is a reflection of the efforts that you guys have been able to put in. And I want to thank the MPS staff for the permits that have allowed us to do that, both at Carlsbad and at Wing Cave. All right, thank you.